first, an introduction to our test subject. This is the Oreochromis niloticus, also known more commonly as the Nile tilapia. If you look here on the left-hand side of the screen, these are the juvenile fry that we worked with. They were approximately an inch in length and had no statistical difference from the 0.5 gram per fish weight across all the tanks. This is what they look like as an adult. They can get up to 2 feet in length, and their weight really varies depending on the fillet size. Now, Nile tilapia are endemic to Africa, and they're grown commercially in Egypt, Thailand, China, and the United States. They're currently one of the 10 most popular aquatic food animals, and the reason for this is they're considered to be very healthy. Their nutritional information per ounce is 96 calories. They have 0 grams of carbs, 1 gram of fat, 7 grams of protein. They're really low in sodium, high in niacin, phosphorus, B12, and selenium. Now, to raise large amounts of aquatic food animals, we use aquaculture. And this is just raising them in these large pond systems. And traditionally, they're fed really high protein feed to maximize their nutrition efficacy. Aquaculture has several drawbacks. One is the crowded conditions. We're trying to grow large amounts of food animals in a very small space. This can lead to infections, in particular antibiotic-resistant infections, and poor growth rates. Also, smaller fry have a harder time to survive, and the survival of the smaller fry is critical. If we need them to survive when they're small so that they can grow up to be big. If we have really high death rates among those small susceptible fry, we're not very efficient. And this limited efficiency is due in part to limited turnover rates. Fry are hatched in what are called as hatch ponds. Little eggs hatch, grow fry. They have to turn into a certain size and weight before they can be moved into growth ponds, and a new hatchling can be brought up in the hatch ponds. So this turnover really limits efficiency. Now, talking about how probiotics and aquaculture are working, this is the main hot area that we're looking at to overcome some of those challenges with antibiotic-resistant infections and limited growth rates. Now, our consensus definition of a probiotic, gathered from various sources, is that it's a microorganism which, when administered to another organism, allows for some advantageous effect. The two most common effects that we're looking for are immune boosting properties and growth enhancement. Now, typically, when we talk about probiotics, we're talking about bacteria. And we're looking at them to work in one of two ways. Either out competition. So the gut surface is limited. It's very finite. If you can get colonization of one bacteria in a space, it won't allow for colonization by another bacteria. And probiotics sometimes just simply take up that mucosal lining and don't allow other bacteria to colonize. The second way that we look at is enhancement. Either we can get an immune boosting property or we can get some greater growth enhancement. Common probiotics to aquaculture in terms of tilapia vary, but there are 3 primary ones that have been studied. Lactobacillus has been used across all different types of aquatic organisms. It's very common to use these lactic acid bacteria, but they're mostly looked at for immune boosting properties, and no studies have been done looking at them to enhance growth. So we're looking in this study to enhance growth, so this one wasn't selected. The second one that was studied is Micrococcus luteus. This one was studied to see if it could improve immune function when a fry was subjected to pathogens, and if it would increase growth. And they did see a statistically significant increase in growth. There was a 7% difference in the final weight at the end of the study between control and treated. And it did improve immune function with some variability. The first study that was done just to enhance growth, not looking at immune boosting properties, is Enterococcus faecium. This one was selected and did show an 11% increase in final mean wet weight in control versus treated. Now, when we are selecting probiotics for aquaculture research, we're looking at 2 different types. There's aerobic and anaerobic. These are the 2 main categories that bacteria fall under. Aerobic bacteria need oxygen in order to metabolize carbon sources. Anaerobic bacteria can do it without oxygen. And within anaerobic bacteria, we're looking at 2 different types. Facultative, which means that they can survive in the presence of oxygen and they can survive in the absence of oxygen. And obligate anaerobes, which do not survive in the presence of oxygen. All of these different types of microbes are common to gut tissue, although anaerobes are considered slightly more common. So we chose Eubacterium cellulosolvens 5494, which is a completely novel bacteria to study for probiotic growth enhancement in fish. It is gram-positive, non-spore forming, it is cellulitic, and it is an obligate anaerobe. Now, the reason why we chose this bacteria is threefold. First, it is a very efficient metabolizer of cellulose. If you remember, we talked about those high protein feeds. They can be very expensive. The filler that is in with the protein is cellulose. This bacteria has the ability to break down cellulose and use it for nutrient sources and release the byproducts of it for uptake by the host organism. This is also, if we can cut the amount of protein in the feed, if cellulose can be utilized by the fry, we might be able to decrease the cost of feed. No member of this genus has been found to be a primary pathogen. So Eubacterium are considered safe. In fact, many terrestrial animals, humans and pigs in particular, have been found to have this particular bacteria in their gut microflora. And the obligate anaerobe status also minimizes the environmental risks. So since this will not survive in the presence of oxygen, we don't have to worry about it escaping the fishery and causing widespread environmental harm. Now, when we're talking about this probiotic, we're looking to see if it can assess growth. So there are two methods by which we assess growth enhancement. The traditional method is to determine mean wet weight before and after a treatment. So you take all of your fry, weigh them using a digital scale, divide it by the number of fry that you have, you get a mean wet weight. That's the traditional method, and previous studies have looked solely at this. The more resolute assessment of growth that is being studied just in fish in general, and we decided to apply to our probiotic study, is to look at real-time polymerase chain reaction utilizing liver tissue to assess insulin-like growth factor 1 expression. Now, to understand about real-time PCR, we first need to do a little micro review of genetics. We know that the central dogma of biology teaches us that DNA is transcribed into RNA, which is then translated into protein. And when we're trying to ask a question about what genes are being expressed in transcriptional expression, we ask that question using real-time PCR. So not to get too complicated here, but to cover the basics of real-time PCR, what we're doing is taking liver tissue and we're extracting RNA, getting all of those nucleic acids together. We synthesize a primer that will do a reverse transcriptase reaction to give us cDNA. We need complementary DNA in order to perform the real-time PCR. The primer sits down on the mRNA transcript, anneals or extends down the line, and then is released, and we end up with a single-stranded cDNA. Okay? We take this through TaqMan-based real-time PCR using a dual-labeled probe, and we standardize all of our expression using beta-actin. Beta-actin is important for standardization because the IGF-1 expression in the mRNA cDNA synthesis reaction can be variable, but beta-actin is the same in every cell within a tilapia. So we use it to set our standard. Now, performing this TaqMan-based system involves using 1 microgram of our total RNA that we extracted from our liver tissue as a template for cDNA synthesis. We then take that same cDNA, 1 microliter of it, and use it as a template for each PCR reaction. The real-time PCRs are conducted using standard PCR mix, and standard curves are generated using plasmid that contains the amplicon of interest, the IGF-1. If you look up here at this, the diagram is a little small. This is our forward primer. It sits down on our cDNA and causes exonuclease activity. When it hits this area right here, which is where our fluorescent probe sits, it causes the exonuclease. The probe breaks free from where it has bound to the cDNA. It also breaks free from this piece right here, which is called a black hole quencher. The fluorescent part comes loose and separates a distance from the black hole quencher, which was keeping that fluorescent from showing up. This computer system right here is capable of reading the level of fluorescence and determining quantitatively how many times we saw this reaction happen. Now, we've been talking a lot about IGF-1. Let me explain to you why that is significant. The growth axis consists of taking hormones from the brain and getting them out to tissues to cause growth. IGF-1 is called insulin-like growth factor, and the reason why is that the structure is actually very similar to insulin, even though it elicits a different response in the cells. IGF-1 causes a cessation of apoptosis, the programmed cell death, and also stimulates cell growth and proliferation. Here, if we look at our somatotropic growth axis, up in the hypothalamus, the body receives signals, varying signals from the body. It sends the growth hormone releasing hormone down to the anterior pituitary gland, which in turn releases growth hormone out systemically. Growth hormone binds to growth hormone receptors in the liver and stimulates the expression of IGF-1. IGF-1 is expressed in the liver, leaves the liver, and goes out systemically to tissues. Connective tissues, such as bone and muscle, will have the IGF-1 receptor, and when the IGF-1 receptor binds to these cells, it causes the cessation of apoptosis and the increase in proliferation and cell growth. The IGF-1 is a really key regulator because it is here, and also these cells can express it independently on their own, but we look at the liver because that is the main hub for this increased growth rate. So for our experimental design, we wanted to make sure that we are asking only one and answering only one question at a time. This is what's really critical about making sure that you have good experimental design. We're trying to eliminate any variable that could confuse the question and the answer. So our null hypothesis is that Eubacterium cellulosolvens 5494 will not affect the growth of the Oreochromis niloticus, as determined by no statistically significant difference in final mean wet weight and expression of hepatic IGF-1. Our alternative hypothesis is that Eubacterium cellulosolvens 5494 will serve as an effective growth-enhancing probiotic when administered to the Nile tilapia, as determined by statistically significant increases in the mean wet weight and expression of hepatic IGF-1. So in an effort to make sure that we're asking and answering only one question at a time, we took 6 tanks, set them up in the lab. They were 10-gallon tanks. We distributed 25 fry into each tank. Each fry was considered to not be statistically significant in weight from other fry. Wet weights were equal across all tanks. And we made sure that we had a good genetic mix, that we were pulling from several clutches of fry. We put them on a timed 24-hour light, 24-hour dark cycle, and temperatures were equalized across all tanks at about 26 degrees C. Each tank was fed the same amount at the same time from the same food source across all tanks throughout the experiment. E-cellulosolvens was grown in peptone yeast media that had been supplemented with a 5 millimolar solution of cellulobiose and was grown to an optical density of 0.5 at 650 nanometers. We extracted 1 mil of the resulting culture, formed a pellet via centrifugation. Then we took it through 2 wash steps with sterile saline. This is to make sure that we're removing any possible culture media. We do not want that carrying over and confusing the question and answer. We mixed it with the same amount of daily feed that they would have received, again from the same food source, and presented it to the fry. At the same time that treated fry were presented with their treated feed, control fry were presented with untreated feed. And this was repeated on days 6 and 10 for a total of 3 doses over 10 days. When we entered the weight collection phase, the fry were removed on day 45 and total wet weights were determined using a digital scale in grams. Surviving fry were counted and mean wet weights were determined, and all data was evaluated using standard t-test and mean statistics. Liver tissue collection was done by removing 5 fry from each of the tanks. We euthanized them using MS-222 at a 0.5% solution, dissected them using a standard dissection kit, and between 50 and 100 milligrams of liver tissue was collected. The liver tissue was stored under tri-reagent in the -80 freezer to protect from RNA degradation until the samples could be processed. The samples were run through the standard real-time PCR procedure that we talked about earlier, and we had a pre-designed primer and probe ready to process these samples. All the collected data was standardized using beta-actin, and results were evaluated using, again, the standard t-test and mean. So for our results, this is our wet weights. We had a final mean wet weight in the control fry of 2.76 plus or minus 0.51 grams, and we had a final mean wet weight in the treated of 3.66 plus or minus 0.29 grams. This is per fry. And if you see here, just as a reference point, this is our initial weights, average weights across tanks, and there was considered no statistically significant difference in weights. All values were considered statistically significant with P as less than 0.1. What's interesting is that our treated fry had a 32% increase over control fry at the end of the experiment. So we had 32% difference in weights. The two previous studies that were done looking at growth enhancement in tilapia, the Micrococcus luteus and the Enterococcus faecium, they saw a 7 and 11% increase in weight respectively. So we saw almost 3 times as much increase in growth versus the other two previous studies. IGF-1 expression data was also considered statistically significant. We saw a total 4.85 plus or minus 0.08 expression of picograms per microgram of total RNA in our control versus 6.31 plus or minus 0.22 in treated. And this represented a 30% higher expression level of IGF-1 in the hepatic tissues between treated and control fry. So while previous studies that have looked at these growth probiotics did not investigate this aspect of growth axis stimulation, we did see almost 3 times as much expression going on, which correlates with the statistically looks like it correlates with the growth. So just to show you what the computer gives us when we run the real-time PCR, this is our graph that it gives us. These arrows here that are equally spaced, this is our standard curve. And all of our data points were within that standard curve. Now what that allowed us that it was a little bit easier to do our statistics. We were able to interpolate our data points rather than having to extrapolate them since all the data was within the standard curve. Now for discussion, talk about the significance of the results. There was a statistically significant difference in growth rate. There was a statistically significant difference in the hepatic IGF-1 expression. So based on this, we reject the null hypothesis and we do not reject our alternative hypothesis. Now for what's next, it would be really interesting to be able to look at where the physiological interaction is happening with the growth axis between the presence of the bacteria and this growth axis stimulation. What physiological processes are happening? How long does this bacteria remain colonized in the gut? Is it something that is transient, just passing through? And would prolonged colonization enhance benefits? We'd also like to see if this protocol works with other aquatic food animals. Would it work, in other fish? Would it work in crustaceans? And how could this be used in the real world of fisheries? So we know that it's an obligate anaerobe. Most fisheries are not going to have the capabilities of producing this large scale while minimizing the exposure of oxygen. So we need to see if heat-killed or lyophilized cultures would be just as effective. And in conclusion, we did determine that the application of the Eubacterium did appear to significantly influence the growth in the Oreochromis niloticus. Now, I would like to take a moment to express my extreme appreciation for all the people that have helped with this. My committee members, Dr. Murdock, Dr. Mead, and Dr. Blair, who have been very patient with me and have worked tirelessly with me on this project. Dr. Romano and the JSU Biology Department, because they've always worked hard to support research and to be a listening ear when science gets frustrating. Dr. Hugh Hammer of Gadsden State is the one that so generously donated all of our tilapia fry, and he also served as an advocate when we had questions about conducting aquaculture research. Dr. Clanton of Jacksonville Animal Clinic actually gave me syringes to help make this media, which I cannot express the amount of help that was. and Jacksonville State University in general for providing such an incredible education atmosphere that made me want to stay here and work on this project and graduate again from Jacksonville State. So thank you, and at this time, if you have any questions. Any questions? Yes, Dr. Aldenay.
How many times do you repeat the experiment?
This experiment project and was successful, and in that attempt they used heat-killed bacteria. I repeated the experiment for this research. The experiment was repeated one more time, but we had an issue with temperature control in our tanks, and our fry passed away.
So that's a combination of what?
We had 3 replicates per tank. So even though there were 25 fry in a tank, we counted that as 1 replicate. Is that what you're asking?
P-value 0.1 or 0.01?
It's 0.1. It's considered, it's not the best p-value, and actually, but in aquaculture research, it's considered statistically significant at 0.1. That's kind of the standard because fish are so.
0.05 is the standard?
Well, 0.1 is acceptable in aquaculture just because of the high rate of demise of fish. They're so susceptible to things that can kill them off. In literature, of aquaculture research, it is acceptable to have a 0.1, although it's not ideal. Our IGF expression data actually was 0.05, but we wanted to be consistent across all results with what we were setting as our standard. Any other questions? Yes, sir.
The IGF, the growth factor you were looking at, your numbers were in picograms per...
Picograms per microgram of total RNA. So we used the spectrophotometer to determine our concentrations prior to doing our cDNA, so that we made sure that we were using equal amounts.
Well, my question is there other data out there on other fish species that have looked at that growth factor? And
Yes,
what are the numbers, what do those numbers compare, I guess?
Well, the studies that were done were to determine if IGF-1 expression in the hepatic tissue was a legitimate form of correlating with growth axis stimulation. So that was reported in the same values, I guess. But nobody has used this to look at giving something to fish and determining if it's affecting growth axis stimulation. This is very novel research in that regard.